Yep, I'm Rachel Simister. I'm a research associate in the departments of microbiology and immunology and Earth, Ocean, Atmospheric Sciences at UBC. And today I'm going to talk about how you can use DNA from microorganisms as sensors for concealed mineral deposits. And so, yeah, Stephen just gave us a great intro in all the wonderful things that you can do with DNA from microorganisms. And yeah, I wanted to kind of reiterate that the kind of thought behind using microbes for mineral exploration came from the fact that, yeah, microorganisms are ubiquitous in almost every environment on Earth. They're highly sensitive and responsive to chemical and physical changes in their environment. And so together, they're largely responsible for catalyzing biogeochemical cycling on Earth. And so I find a really great way to sort of visualize this is to look at phytoplankton, microorganisms in the ocean, responding to trace amounts of iron. So these are like trace concentrations of iron that change, and you can visualize these microbes from satellite imaging from space. And so if we take the same principle through to soil, in a gram of soil, it's estimated that there can be as many as 10 million living organisms, and these comprise tens of thousands of bacterial species, each sensing and interacting with their surrounding soil environment. And so historically, microbiological techniques have tried to be used in mineral exploration, but they'd found limited application. And that was because some of the methods Stephen was talking about, that we didn't really have the technology until very recently, and sequencing used to be quite expensive. I've just got a couple of slides to illustrate that. So this is what the cost of the human genome, how it's changed over time. So if you go back to the early 2000s, you're talking of tens of millions of dollars to sequence the genome. Whereas now we're looking at potentially even like $1,000 to sequence the genome. And then coupled with that, we've really had a sequencing revolution, if you will. So the amount of data that we can now generate is, we've got billions of base pairs of data. So back in the early 80s, you might be producing 10 kilobases of data per day, whereas now, yeah, we've got lots and lots of data. And that means that we actually now have high throughput techniques that can rapidly and cost effectively capture sequence data from the environment, and we can apply this to microbial community information during mineral exploration. So Canada is a great place for us to test this idea. And that's because, as we know, many deposits are being mined out, and we're having to often find deposits that are sort of undercover. So we're trying to see through overburden, if you will. And so Canada is a great place to, test some of these exploration undercover, if you will. So we've been looking at 4 different sites across Canada. So we've got 2 kimberlite sites in the Northwest Territories. We've got a Kelvin kimberlite, a DO18 kimberlite, and we've got 2 porphyries in BC that we've been looking at. We've got a copper gold porphyry and a copper molybdenum porphyry, and just big thank you to all the government and industry sponsors that have taken part in this project. And so just wanted to kind of show you a few slides on like, what does a microbial mineral exploration look like? And so it's quite similar to if you went out and did a geochemical exploration. So you would have initially sort of found the area that you want to explore. You would do some transects. You dig your holes. In this case, we always take B horizon soils across our transects because we want to be consistent across where we're looking. And essentially, you'd bag a sample for bulk geochemical analysis. We just need an extra 200 grams of soil for microbial analysis. So it's quite typical to what you would normally go out there to do. And then when we come back to the lab, we basically extract the DNA. This DNA then represents all the microbes that are in the soil. There's many different things, as Stephen just went over, that you can do with this DNA, but we're just focused on the barcoding part, the finger parting part. So we're just looking at one gene, and that's the 16S rRNA gene. That tells you which species of bacteria are in your sample and what's their relative abundance across the area that you're surveying. So I'm going to just show you a couple of plots from our kimberlite sites, just so you can see, like, if you were to do this kind of work, what would you actually end up as? What would the data look like? How would you define your drill target? So we're looking at the DO18 and the Kelvin deposit. So the first one, I'm going to show you what typical chemical data would look like. So this here is our DO18 deposit. The black is just showing you the outline of the deposit, and these bubble sizes and colors that you see as the concentration of the chemical element. So in this case with the chemistry, we're looking at 4 pathfinder elements: chromium, magnesium, niobium, and nickel. And as you can see from this map, they were found to be spatially associated down ice. So this is because of the mechanical glacial dispersion, they're often down ice. Whereas when we do this with the microdata, you'll see that we get a much clearer spatial correlation with the surface projection of the kimberlite. And we can plot our microbial data the same way that you would look at a chemical anomaly. So these bubbles are showing you an increasing shift in the relative abundance of the microbes. And the next thing that we can do is just sum these. So I'm just showing you the comparison between the chemical fingerprint on the bottom and the microbial fingerprint on the top. And so the thing to remember though is that with the chemical fingerprint, you've only got 4 elements here. With the microbial fingerprint, this is made up of like tens to hundreds of different species, and it's only 1 gene. So you could imagine if you start to use more genes, your statistical power of defining a drill target greatly improves. And so we thought, well, we should try this at another site just to, you know, see if it works again. So this is the Kelvin site. And once again, you're looking at the kimberlite projected outline is in the black. And then you've got these bubbles, which on the chemical one shows increasing concentration of the element. With the microbial fingerprint, it's increasing relative microbial abundance. And as once again, what you're seeing is that the microbial fingerprint more clearly defines and resolves the buried kimberlite. And the thing about both these sites to remember is that with DO18, it's under 5 to 10 meters of overburden. And with this Kelvin, it's buried 150 meters down in the bedrock, and it only outcrops actually under the lake there. So we're actually seeing through overburden. And so just to kind of round off of the things that we're kind of interested in. So now that we've shown that, you know, it seems to work well, we've also done this in BC. We're actually looking at how can we make this better so that it can be used by industry. And so we're looking to develop simple and robust field sampling protocols and strategies. We're looking to build databases of background microbial communities. So that's how we get our anomalies, right? We're comparing the community over the deposit to the background. And so by having more background, a better idea of what that is, we can better define our anomalies. And so we're looking to do this in specific soil types and terrains. As I mentioned, this data is based on 1 gene. We're looking to expand the number of sensors we have, increase our statistical power, so develop more sensors, more functional genes. And then we're looking at integrating this with geophysical approaches. So maybe you would initially do your drone-based magnetometry and then hone in using this microbial fingerprinting technique. And so yeah, if anyone's got questions or wants info on some of these things, you can contact myself, Sean Crow, Craig Hart, and obviously BRIMM. And that's all I have.